Today, we are going to review the structure of DNA. DNA is the master copy of our genetic information that holds the code for making proteins. The monomers for DNA are called nucleotides. There are 3 parts to a nucleotide: a 5-carbon sugar, a phosphate group, and a nitrogen base. The 5 carbons on the sugar are numbered 1 through 5 clockwise from the O. The phosphate group is attached to carbon number 5, and the nitrogen base is attached to carbon number 1. This particular nucleotide has the thymine nitrogen base attached, so the name of this nucleotide is thymine. Nucleotides attach to one another in a long strand of polynucleotides. The bond that holds these nucleotide monomers together is called a covalent bond. The order of nucleotides on this strand is T, G, C, A. Given this sequence of nucleotides, write the complementary sequence. That is correct. A always pairs with T, and G always pairs with C. So if we know the sequence of one strand, we can predict the sequence of another. The bond that holds these 2 complementary strands together is called a hydrogen bond. You'll notice that the complementary nucleotides appear to be upside down. The phosphates are facing the bottom. The term for this arrangement is anti-parallel, which means if we were to count the carbons on the left, carbon number 5 would be on the top and carbon number 3 would be on the bottom. The complementary strand is anti-parallel, which means carbon number 3 is on the top and carbon number 5 is on the bottom. So when we write a sequence of nucleotides, it's important to show the direction. And when we write the complementary sequence, it is important to show the anti-parallel direction. We are now going to learn how DNA makes a copy of itself in a process called DNA replication.